Hi, ladies and gentlemen. I just wanted to walk you through how to do your pasta genetics lab activity that you're going to be working on. Hey, I would say right now would be a good time for you guys to go through and read a little bit about inherited traits or the inheritance of traits. Okay, so right here it says genes are located in the chromosomes of cells with each chromosome pair containing two variants of each of many distinct genes. Whew, right? So when we talk about genes, right, the genes are found on chromosomes. Okay, those chromosomes are found in the nucleus, and we have a pair of them. Okay, and when it says two variants, that means that we have two copies of each of those chromosomes, one from our mom, one from our dad. Okay, and when we talk about a chromosome and the genes are going to control the production of proteins, and then proteins get put together to give us our particular traits. And we'll get to that later this unit. Okay, variations of inherited traits between parent and offspring arise from genetic differences that result from the subset of chromosomes inherited. Okay, so again, this is the idea that through the process of meiosis, we are going to get a unique combination of genes in the sperm cells that are created and a unique combination of genes in the egg cells that are created. And therefore, the offspring from a mother and a father are going to have a unique combination of genes. That means there are going to be genetic differences from parent to offspring, right? So we don't have clones or exact copies like we see in asexual reproduction. Okay, so what are our objectives for this lab? I'm hoping you guys learn that genes are passed from one generation to the next, that we have two copies of a gene, okay, and that a child will receive one copy of each gene from each parent, and that which copy of a parent's gene the child receives is random, okay, and that a child won't look exactly like his brothers and sisters because they have unique combinations of genes that form in each gamete. Okay, and identical twins look alike because they have the same genes. Okay, so those are some of our goals for today. Okay, in the next couple of slides, okay, is a review of meiosis. We did this last week, so I'm not going to spend a lot of time on it today. But this is something you can always come back to, especially when we get to our conclusion questions at the end of our unit. Okay, from here, hey, there are two main laws when we're looking at meiosis. And the two laws are the law of independent assortment and the law of segregation. Okay, the law of independent assortment, okay, is going to tell us that when our chromosomes line up in metaphase one, okay, those chromosomes can line up like in this picture, okay, like it's shown on the left or like it's shown on the right. Okay, and when we look on the left, you'll see that we have the blue chromosomes on one side and the red on another. Okay, and we talked about this a lot last week. The reverse could also happen. Okay, so when we look at this alignment in metaphase one, okay, instead of having blue chromosomes on the left, we could have the first pair with the blue chromosomes on the left, but the second pair could line up differently. And that's the law of independent assortment. And if we look at the end result, okay, the sperm cells or egg cells that result at the end, this is the same parent, and these are different combinations of genes that are possible. And the reason why they're different is due to that law of independent assortment, is due to those pairs lining up differently. Okay. The second law is the law of segregation. Okay, the law of segregation is associated with metaphase and anaphase 2. Okay, in metaphase, anaphase 2, the individual chromosomes that line up, those individual chromosomes are going to get pulled apart so that in the sperm cells or the egg cells that form, we have unduplicated chromosomes. Okay, that means the sister chromatids segregate or separate. Okay, and so that's why we have unduplicated chromosomes in the cells that result from meiosis. Okay, and hopefully we remember that the cells that result from meiosis are haploid. The cells we started with were diploid. Okay, diploid meaning having 2 copies of every chromosome. Haploid meaning having half that, so only 1 copy of every chromosome. Okay, from here, let's take a look at our procedure and then I'll kind of walk you through. So I have a written procedure here, and then here's the actual lab steps or where you're actually going to perform those procedures. Okay, so you're looking at slide 4 and then you move to slide 5. Okay, so in slide 4, it says you have 8 pieces of pasta, 2 of each shape, and they're placed in each cup. Okay, the pasta shapes stand for chromosomes. There are 2 of each pasta shape because we have 2 copies of each chromosome in our diploid cells. Remember, the shape of the pasta is the chromosome, okay, each containing different genes coded for by the DNA, and the color of the outline, okay, and the color of the noodle helps us know where we got that gene from. Okay, is it maternal from the mother? Okay, or is it paternal from the offspring's father? Okay, so how this is going to work, randomly choose the chromosomes for the mother. Okay, this is the daughter of grandfather 1 and grandmother 1. Okay, how we're going to use that is we are going to use the coin flip link, okay, at the bottom of the slide. Okay, so you'll see this, I'll show you on the next slide. Randomly select the 4 chromosomes from grandfather 1. Okay, make sure that you take 1 of each shape from grandfather 1 and then repeat for grandmother 1. Okay, again, make sure there's 1 of each shape and those pasta pieces are going into the mother's cup. Okay, so that's like going into creating the zygote, which will then develop into mom. Okay, so the mother's cup are mother's chromosomes. Half came from her father, which in this case is grandfather 1, and half came from her mother, which is grandmother 1. Okay, so let's look at actually doing that. Okay, so I am now on the next slide, so slide 5. Okay, on slide 5, you'll see these are grandfather's genes. Half of them are outlined in green, half of them are outlined in purple. Okay, they're all the pasta shapes are brown. That's going to tell me they came from grandfather 1. Okay, so we need to remember that when we get to the end of the lab. Okay, the reason why there are some outlined in purple and some outlined in green, the purple outline says this came from grandfather 1's mother. The green ones came from grandfather 1's father. Okay, so what we're going to do is you'll see we have 2 like penne noodles. Okay, do we want the green one or the purple one? Okay, so that's what we're trying to decide. I'm going to use the coin flip app. Okay, and if it lands on heads, I'm going to take the purple outlined penne noodle. And if it lands on tails, I'll take the green outlined one. Okay, so I'm going to click on this link. Okay, and so you'll see that this has a little flip link here. Okay, so from here I'm going to hit heads or tails. Okay, it spins around, it's going to land on something. It was heads. Okay, I'm going to go back to my pasta noodles here. Hey, heads, hey, has to do with the purple outline. So I'm going to come up here and I'm going to take the penne noodle with the purple outline, just the penne one. Okay, because remember, each one is selected independent of the others. Okay, so then I'm going to go to the bow tie noodles. Okay, I'll go back to my simulation. I flip. Okay, this time I end up with tails. Okay, so for the penne or for the bow tie noodles, tails is the green outline. So I'm going to grab the green outline one and I'm going to drag it down, okay, into mom's cup. Okay, then I move on to my shells. Okay, I flip and I get tails. Okay, since I've gotten tails, again, tails is the green outline. So I grab the green shell. Okay, and I don't care if it's truly like right in the cup or if it's just near the cup is fine. Okay, it's just the idea that, right, that it's going into and it's going to be what becomes mom. Okay, then we got our elbow noodles, same thing. Okay, we flip again. Okay, it landed on tails again. Okay, so tails was the green outline. So I grab my green outline for the elbow noodle. Okay, now I have one of each noodle type from grandfather. Okay, so remember these are representing chromosomes. This is like chromosome 1, 2, 3, and 4. Hey, we get one copy from grandfather and then one copy from grandmother. Okay, so I'm going to follow that same procedure with grandmother. Okay, so again, I'm going to go back to my simulation. Hey, you might speed this up here. I've got tails. Okay, so grandmother 1, if I'm looking at the shells, tails was green outline. Okay, so I'm going to bring down that one. Okay, then I'm going to go through, do it again. Okay, I ended up with heads. Okay, so for the elbow noodle, I got the purple one. Move that one down. Okay, and you'll notice that all of these are blue. The ones that are blue came from grandmother 1. Okay, I'm going to pause while I continue my flips, so hang on a second. All right, so now you can see, hey, mom has received 2 copies of the penne noodle. Okay, one from grandfather 1, one from grandmother 1. 2 copies of the shell noodles. Okay, one from grandfather 1, one from grandmother 1. Same with the bow ties, same with the elbows. Okay, there are 8 chromosomes, okay, in mom. Okay, and if we go back and look at the grandparents, hey, the grandparents started with 8 noodles, hey, as well, right? So the number of chromosomes we start with, we end. And what we're showing here, right, by combining them in the cup, that is the idea of these brown noodles all together represent the sperm. Okay, the blue noodles all together represent the egg, and then they fuse together in fertilization, hey, to make a zygote or to make a baby. Hey, we would follow these same procedures for grandfather 2 and grandmother 2. Hey, so again, I'm going to pause here so you don't have to watch all my flipping. Hey, just to get my noodles. Do this yourself. Hey, don't take mine. Otherwise, we are going to end up with bad data. Okay, so each person's has to be random. Okay, so we'll pause here for a moment. You should do the same. Hey, fill up your mother's cup and your father's cup so you know what to do next. Okay, I've now filled up my father's cup as well. Okay, so I now have, hey, again, 1, 2, 3, 4, 5, 6, 7, 8 chromosomes in the father. Hey, 2 copies of the bow tie chromosome, 2 copies of the penne, etc. Okay, so when we talk about having 2 copies of something, hey, the father is diploid, the mother is diploid, hey, and then we are going to go through the process of, hey, them having children. Okay, so if this mother and this father have 4 children, hey, what combinations of genes could this child get? Okay, and this is where it's really important that you're using this coin flip so that we're getting random combinations. Okay, once you've completed that, hey, and you have filled in your cup for your child, hey, you're going to screenshot or take a picture of your cup filled with your child, hey, and you are then going to copy that picture into here. Hang on a second. So, hey, you will fill in, hey, and again, it's random, so make sure you're using the coin flip, hey, to select, hey, random chromosomes from mom. Hey, mom will give 1 copy of each chromosome. Hey, otherwise, right, our child will have, hey, so a chromosomal disorder. Hey, father, okay, will also give 1 copy of each chromosome. Okay, so make sure again you're using the coin flip so you have a random combination of these genes. Okay, so you know what combination, hey, we've got. Okay, so this is my first child. Hey, the first child from mom and dad. Okay, so what I'm going to do here, hey, I'm going to use my snipping tool. Hey, you guys could do a screenshot or something like that, but I really just want the child. Okay, so I'm going to take that. okay, and get a little picture of it. Okay, then I'm going to copy that and I'm going to go to my next page. This is my first child from mom and dad. Okay, I'm going to move that child's cup so that it fills in this little space. I'll resize it so it fits in here. Okay, and once you've done that, you're going to name your child. Okay, so you put a name here, and then you're going to tell me, hey, how many chromosomes originated from grandfather 1? How many originated from grandmother 1, grandfather 2, grandmother 2? How many total genes are there? And how many total gene pairs are there? Okay, so remember, grandfather 1. Okay, grandfather 1 had brown chromosomes. Okay, there are 1, 2, 3 brown chromosomes in the child, so 3 came from grandfather 1. Okay, originated from grandmother 1. Okay, grandmother 1 was blue. There is 1 chromosome that came from grandmother 1. Originated from grandfather 2. Okay, grandfather 2 was yellow. There are 2 chromosomes that came from grandfather 2. Okay, and how many chromosomes came from grandmother 2? There were also 2. Her chromosomes were yellow. Okay, so when I look at this cup and the chromosomes, you'll see we have 2 copies of the shell chromosome, 2 copies of the bow tie, 2 of the elbow, right? This child is diploid. Hey, how many total genes or really chromosomes? Hey, there are, 8 chromosomes here. Hey, how many pairs of chromosomes? How many homologous pairs are there? Okay, so if we take a look at the homologous pairs, there are 4. Okay, you're going to follow that same procedure, okay, from mom and dad down to child, okay, until we have 4 different children. Okay, 4 different children. Okay, so the key is, how are we going to do that? Well, I'm going to return all these chromosomes that the child has. I'm going to return them back to mom. Okay, return the chromosomes back to dad. Okay, fill up mom and dad's cup again. Okay, because every time a child is born, okay, or every time a child is conceived, okay, that's due to meiosis occurring, okay, and it's a new, okay, process of meiosis. So we kind of go back to our original cell. Okay, so we have our original cell here, okay, and this original cell for the father and the original cell for the mother. Again, random, which copy of each chromosome gets passed on to the child. Again, take a picture, okay, fill in, put your picture of the child's cup here, and then fill in the chart, okay? So that's what you guys are going to be working on today. Once you finish that up, okay, there are some conclusion questions and a comparison to the real world. Okay, I'm going to have you guys fill this out. When you guys are filling this out, these conclusions, okay, how many, okay, these are pretty simple questions, right? How many genes, hey, did the child receive? How many of each pasta type? Hey, did any of the children have the same combinations? Why or would this not happen in humans? Hey, did every child get at least one of his or her genes from each grandparent? Hey, in this simulation, we have 4 chromosome pairs. Hey, would you expect the same genes to be found on each pair? Okay, so remember, like the pair would be like the 2 penne pastas. Okay, and then, hey, over here, you need to tell me, is it or is it not? Okay, possible. Is it or is it not? And then explain why. Okay, is it or is it not? And then explain why. The next one, hey, this is comparison to the real world. Hey, you are given 5 different options. 1, 2, 3, 4, 5 different options. You're going to tell me which one of these is like fertilization. Which one of these is like the zygote being formed? Which one of these explains independent assortment or is related to independent assortment? Which one is related to segregation? Which one is related to a chromosome? Okay, and So it's like the father's cup is like, hey, the father's cup is like a zygote, is like fertilization, is like independent assortment. So you're picking which one it's like, and then you say because, and you need to include characteristics of both parts. Okay, so the father's cup has chromosomes that came from grandmother and grandfather two. Okay, and that's like which one of these because and explain why. Okay, in this case, it would be like a zygote. Okay, what characteristics or traits do zygotes have? Okay, so that's what you guys are going to be working on today. Please let me know if you have any questions. Send me a chat message. Sorry I can't be here with you guys this week, and I hope it all goes well. I'll talk to you soon.